Hello, students. Today we will be reviewing osmosis, tonicities, and active transport for today. We're going to go ahead and start with a video review on osmosis. If you don't remember what osmosis is the type of passive transport that only works with water molecules that are going to be going through the membrane. Remember, the different types of names for your membrane include cell membrane. That's one of them. The second name we have is plasma membrane. The third name is the phospholipid bilayer. And the final name that we name the membrane, which they have like a little picture there that they're showing you, is the fluid mosaic model. So we're going to be exploring how osmosis, which is the type of passive transport that works only with water molecules, helps me pass those water molecules through the membrane and the many names that it has. So remember, guys, that passive transport is a type of cell transport that does not use energy or ATP. And so it's going to help me pass my water molecules easily since water technically does not have a shape. Now, sometimes we might have some special proteins that are usually embedded or in the middle of the membrane called aquaporins that'll help me transport my water molecules. But let's go ahead and look at our little video presentation here. And we're also gonna be covering tonicities. The three tonicities that they're gonna mention right now, and I'll stop when they get to that part, are hypotonic, isotonic, and hypertonic, okay? And then I'll go ahead and stop at the examples when they show them. Let's go ahead and look at this first.
Diffusion is the movement of particles from an area of higher concentration to an area of lower concentration. Diffusion occurs because of the random movement of atoms and molecules that is referred to as Brownian motion. Diffusion can occur in liquids, gases, and some solids. If food coloring is dropped into a beaker of water, it will gradually diffuse until the color is equally distributed and a balance is reached. This state of balance is called dynamic equilibrium. The rate of diffusion can be affected by concentration, temperature, and pressure. An increase in any of these factors will increase the rate of diffusion. Osmosis is a specific kind of diffusion. It is the diffusion of water through a semi-permeable membrane such as the cell membrane. The cell membrane, also called the plasma membrane, only allows certain molecules to enter or leave the cell. It is said to be selectively permeable. During osmosis, water moves from an area of higher water concentration to an area of lower water concentration. In other words, water will move across the membrane into the side that has the lowest percentage of water and the highest percentage of solute. In this example, water will move into the cell, and the cell will swell.
So pausing right here really quick on this example. So here, if your biology teacher gives you some problems that kind of have some percentages like this, it's probably not going to have like a little cell. They're probably going to give you some problems that have like little beakers with a little cell in it. So the way to do those, guys, is that, for example, let's look at the cell on the left right here first, okay? So during your osmosis problems that your biology teacher is going to give you, they're usually going to have percentages. You're going to have 2 over here on the inside of the cell, and you're also going to have 2 outside of the cell. You're usually going to have H2O or water, and then another solute, maybe like sugar or salt or something else, okay? Every time we're working with these percentages, they have to add to 100%. So for example, 96 plus 4, 96, 97, 98, 99, 100, right? That's 100% outside of my cell right here. So that's perfect, right? And then on the inside, you have 92 plus 8, so 92, 93, 94, 95, 96, 97, 98, 99, 100. Perfect, right? So they're both 100%. So as long as both of them equal to 100% both in the inside and the outside of the cell, I'm fine, right? I'm good. So that's that for the percentages. Now, every time I'm gonna have more, let's say here, you look at the percentage, sorry, that has more water. So I'm gonna compare my two percentages on the outside and the inside of the cell. So on the outside, it says I have 96% of water. And then on the inside, it says I have 92% of water. So since I have more water on the outside of the cell, that's why over here on the right, they're saying that the water is going to be going into the cell. You see that? And because the water is going into the cell, it's going to get chubby, right? Or it's going to expand, all right? So because it's going to get chubby or expand, I like to say this is hypotonic or hippotonic, right? Hippo, chubby, right? Chubby like a little hippo. So this is one type of tonicity that they're showing you here. And they're usually, again, they're going to maybe show you a little cell. They might have like a little beaker around it. It'll have the percentages inside of the cell and outside of the cell. Remember I said they're supposed to add up to 100%. And again, like for example here, since I have more water on the outside of the cell, in order for it to kind of even out, in order for them to achieve homeostasis, that means maintaining a balance, or they might call it an equilibrium, the water or the extra water that I have on the outside of the cell needs to come into the inside of the cell so that it can be balanced out or reach that equilibrium or that homeostasis. Okay, let's keep going.
The terms hypertonic, hypotonic, and isotonic are commonly used when comparing a solution to the contents of the cell. The terms can also be used to compare any two solutions. When a cell is in a hypotonic solution, there is less solute in the solution than inside the cell. Water will move across the membrane and into the side with the greatest solute. As water moves into the cell will expand or swell. Animal cells may continue to swell until they burst.
So here's another example of a hypotonic or a hippotonic solution, right? So you're gonna usually have less solute, which is maybe like sugar or salt in the solution or the water. That's what they mean, right? So for example, I would have less water or solution right here. And then they're saying I'm gonna have more water inside of the cell. So that's why, for example, here I have the arrows pointing towards the cell. And if I don't know if you can tell, but it says H2O here in all of my little arrows. So a lot of the H2O is going to be going into the cell. That's why the cell swells up or gets chubby like a little hippo, hippotonic, hypotonic, right? And then sometimes they can rupture or burst like a balloon. Okay, so this one is hypotonic.
When a plant cell is in a hypotonic solution, it will also swell. However, the cell wall will prevent the cell from bursting. The pressure that results from the movement of water into the cell is called turgor pressure. When a cell is in a hypertonic solution, water will move out of the cell, and the cell will shrink.
So this next tonicity, it's telling you there, it's called hypertonic, right? The other one was hypotonic. Hypotonic, hippotonic, the cell's getting chubby because all the water's going into the cell. This one is opposite, right? Here they're showing you the little green arrows pointing out of the cell, right? So that means all of the water is going to be going out of the cell. So if I only have, let's say, salt or sugar left in the cell, it's going to shrivel up like a little raisin, right? So that's why you saw, for example, right now, the cell is going to shrink up like a little raisin, okay? Because all the water is going to be taken out of the cell and it's going to go into the solution or the water that's surrounding the cell in the little beaker. So this is where I have water moving out of the cell and then the cell will shrink like a little raisin.
As plant cells in a hypertonic environment lose water, the plasma membrane shrinks away from the cell wall. The loss of water results in a drop in pressure and the plant will wilt. When a cell is in an isotonic solution, water moves into and out of the cell at the same rate, so the shape of the cell does not change.
So this is the last tonicity. This one's called isotonic. This is the only one that starts with an I, by the way. So isotonic, if you notice here, for example, the little arrows again, I have arrows going out of the cell. So that means my water is going out of the cell, but at the same time, it's also going inside of the cell. You see that? So because I have my arrows going in the cell and out of the cell, nothing's going to happen to the cell. It's going to stay the same shape because the water is going in and out, in and out, in and out, right? It's going both ways at the same time. So it's not just going out like hypertonic and then the cell's going to shrink, and it's not just going in the cell where the cell expands or gets chubby, hypotonic, hypotonic, right? It's going to go in and out both at the same time. So it's going to be perfectly balanced. The cell shape should not change. It should stay the same, or you're going to have an equal solute and equal water, right? Everything stays the same.
Osmosis is a form of passive transport, which means that no cellular energy is required for the movement of water molecules into or out of the cell.
Okay, so that concludes for osmosis and the three tonicities. Let's go ahead and now go over active transport right here in the notes. Okay, so active transport is totally opposite of what we just covered. What we just covered with osmosis and the three tonicities, which were hypertonic, hypotonic, isotonic, that's all passive transport. That's, again, using no energy to pass through the cell membrane or the plasma membrane. And it's going to go from a high to low concentration. Remember, high to low, going with the flow. Okay. Now this one is going to be opposite of what we just covered. Like I said, this is called active transport. So it tells you there, active transport is the process of molecular movement, which energy is required. Okay. So if I go down here, they are introducing us to two types of active transports. They both start with an E. The first one is called endocytosis. That's where I'm going to have molecules are going to be going into the cell. Endo means inside, going inside of the cell. And then the other one is called exocytosis, being exiting the cell. It's leaving the cell, and both of these are going to be using energy. Remember, another term for energy is ATP. That stands for adenosine triphosphate. ATP is energy. Don't forget. So here it gives us two little definitions. Let's go ahead and look over them really quick. Endocytosis is the action of the cell membrane that surrounds outside materials, and it's fusing with itself to bring in materials. Right now, I'll show you an example of that. So endocytosis, bringing in molecules from the outside of the cell. Exocytosis, exiting, right? So this is the action of a vacuole of a cell fusing with the membrane to expel materials or get them out, right? Or pushing them out of the cell. Now, what's the purpose of this passive and active transport that we're talking about? It's to achieve homeostasis. What is homeostasis? It's the ability of the cell membrane to maintain a stable internal environment or conditions that are independent of the conditions in the environment. So they're doing this to maintain balance, okay? So that our cells and ourselves right here maintain a balance and that we stay perfectly fine. So for endocytosis, remember I said that's the type of active transport that's going to use energy to bring in molecules. Now there's three types, okay? So the three types, the first one is called phagocytosis. This one is also called cell eating. So the cell's going to be eating, let's say, solid particles or molecules. So the way it does it is, let's say it's trying to take in these molecules here. The molecule is going to fuse or attach itself to the plasma membrane here. right? This yellow thing here. Then the cell is going to create like a little pouch, what they call a vesicle right here. And then it's going to take in that molecule by cell eating or phagocytosis, this first one right here. The second one called pinocytosis, this one likes to work with liquid molecules, liquids, okay? So let's say this is another type of liquid. It's going to, again, attach itself or fuse to the plasma or the cell membrane here. It's going to create a little vesicle or pouch and going to be taken into the cell, right? So far, everybody's being taken into the cell. It's just that this one is with solid particles, or molecules. Pinocytosis is working with the fluid or like the extracellular fluid that they see here. And then the last one, receptor-mediated endocytosis. This kind of like works like an enzyme. If you don't remember what an enzyme is, remember I'm going to have the substrate and the enzyme that match up like two puzzle pieces together, okay? And it's very picky. They need to match up together. So this is the way this receptor-mediated endocytosis works. So for example, if the square molecules try to match up with this circle or half circle receptor, they're not going to fit really good. But look at, for example, the star molecules, these fit a little bit better. So these will be able to attach to the receptors on the plasma membrane here of the cell. They're coming from the outside. And then it's going to create like a little vesicle again, or a little pouch around it, and it's going to be taken in to the cell and be used, right, for whatever reason that the cell needs it for. So here are some quick little GIFs showing you how endocytosis works. This is endocytosis in general. The molecule is going to be taken into the cell, create a little vesicle or pouch around it, gets in, and then it gets used. So you can think of this one kind of like your phagocytosis. It looks like a solid particle or molecule, right? Here's another example of phagocytosis here. So these clear looking things are the cell, and they're eating up the bacteria, guys. So sometimes our body does eat up pathogens like the virus or the bacteria to defend us, to help us, right? Now, this is exocytosis. Exocytosis, remember, this is opposite. So instead of taking in the molecules, it's going to take it out. Now, there's only one type of exocytosis, which is good, right? That way we won't have to get bombarded or overloaded with a lot of information. So, for example, here, there's some blue molecules that the cell doesn't need anymore. It's like, I want them out, right? So what they're going to do is they're going to create a little vesicle or a little pouch around those molecules that are not needed by the cell anymore. They're going to fuse together or come attached to the cell membrane, the yellow part here. That's why it says the vesicle fusing with the cell membrane, and then they're just going to let them out, right? Bye-bye. See you later, okay? So here in this GIF here, you have, again, the little vesicle forming attaches to the membrane, and then it's released out of the cell. See you later, right? The last type of active transport we have here is called the sodium-potassium pump. Now, this one is going to be using a little purple protein that we have here. It's embedded or in between the cell membrane because the sodium and potassium ions or molecules are way too big, guys, to pass through the membrane on their own. They're too big. So step number one, it's telling us here, the sodium-potassium pump is going to bind 3 sodium ions right here, these 3. The ones that say Na or Na, right? So these are my 3 sodium ions or molecules. They're going to help bind to the protein pump here. And we're also going to be using ATP, which is energy. Remember that, right? So this is another type of active transport. Step 2, I'm going to have the splitting of ATP. So when the ATP splits apart, that forms something called ADP right here. So this is going to change the shape of the channel, which is right here, so that my 3 sodium ions go ahead and fit in my little sodium pump here. And then the sodium ions are going to be driven through the channel, or they're going to go through the little protein pump, and they're going to get across into the outside of the cell membrane. Step 3, the sodium ions are going to be released to the outside, right? They finally made it to the outside of the cell because they went through the protein pump here. And then in return, I'm going to have my potassium ions or molecules being put into the cell. So they're going to, again, they're too big. So they're going to have to go through the little protein pump here. And then eventually they're going to go into the cell. So this is an exchange of sodium going out and potassium going in. Okay. And again, reviewing both passive and active transport. Remember, passive transport like osmosis and the three tonicities, that one does not require energy or ATP. Usually your passive transport, it's going to go down the concentration gradient. What does that mean? That means it's going to go from a high to low, going with the flow concentration, high to low, right? Remember, high concentration, if I were to be right next to you in the classroom and I spray a cologne or a perfume and we smell it really strong, that's high concentration because all my molecules just got released out of the perfume or cologne bottle and we smell it really strong and the molecules are really tightly packed together. But by the time it reaches somebody, let's say across the room, that would be low concentration because by the time it reaches the other person, let's say across the room, the molecules are going to spread out and they're not going to smell it as strong as, let's say, if I were to spray like right here. Okay. So the different types of passive transport, which is osmosis, right? It only works with water molecules. And remember the three tonicities, which were hypertonic. So shrinks like a little raisin because all the water's getting out of the cell, right? Hypotonic or hypotonic. Remember, all the water's going into the cell. So it's going to swell up, expand, get chubby like a hippo, hypotonic, hypotonic. And then you have isotonic, where you have water going in and also going out of the cell, both at the same time. That's why the cell shape for isotonic, when we're talking about osmosis, is not going to change. It's going to stay the same. Another type of passive transport is simple diffusion. Remember, this one only works with small nonpolar molecules. It doesn't specify. It just says, okay, as long as it's small and it's nonpolar, I can pass through my phospholipid bilayer or the cell membrane without any help of a protein. Then for active transport, like the endocytosis, exocytosis, right? And the sodium potassium pump that we covered, all those are active transport. They all are going to require ATP or energy. These, remember, are going to go against the concentration gradient. So again, what does that mean? That means it's going to go low to high, going against the sky, right? Again, remember that example with the perfume, right? When we spray something that's high, it reaches out to the rest of the people across the room, let's say, that's low concentration. So remember for concentration gradients, really quick, passive transport, that one's high to low, going with the flow. Active transport is opposite, low to high, going against the sky, okay? Now, again, the types of active transport, the ones we just covered, remember that's endocytosis, like going into the cell. Remember, there's three types. The first one is phagocytosis, cell eating, pinocytosis, cell drinking, and the last one, receptor mediated, like an enzyme. They have to match up together, the molecule and the receptor, to go into the cell. Exocytosis, that's another type of active transport. Remember, that's leaving the cell, creates a little vesicle or pouch, fuses with the membrane, gets out of the cell, right? So your teacher should have these notes, guys. So if you click on this drag and drop activity, it'll take you to this page. Let me close this really quick. When you scroll down, here's a little Venn diagram reviewing passive and active transport and what they both have in common. So for example, ATP, right? We said that ATP is energy and it's only going to be used for active transport. So I would put here under active transport. Okay. Another one, we can also say requires energy. ATP is energy, right? So I can technically, again, put that one there, right? Osmosis, we just covered it. That's the water molecule one, right? That one, again, remember, is not using energy, so it would go under passive transport. No energy required, passive transport, right? Passive, it's even in the name, right? Exocytosis, exiting the cell, this one is going to use energy, so it's going to be active transport. Diffusion, this is the one where it has small nonpolar molecules passing through the membrane, not using energy, passive transport. High to low, going with the flow, passive transport. This one's opposite, low to high, going against the sky, using energy, active transport. Used in cell transport, it's for both of them, right? They both happen or go through cell transport. Endocytosis, going into the cell with energy, active transport. Protein pumps, they both use them, okay? Movement of water, this is with your osmosis, right? Oh, right here, okay? Helps maintain a balance or homeostasis, that's for both of them. They both help us maintain internal balance. Cell engulfs / eats a large molecule, that's for phagocytosis, that's a type of endocytosis, so that would be active transport. Facilitated diffusion, we can put that with passive transport. Uses the cell membrane for both of them. Goes with a concentration gradient, that would be for passive, high to low, going with the flow. Against it would be active transport, and moves materials across the cell membrane. We can put it right here. Okay, so this is my Venn diagram for passive and active transport, and then also with the similarities between passive and active transport. You will now proceed to your Google Classroom, guys, for your biology teacher. So make sure that you go ahead and finish off any assignments, quizzes, or other activities or assignments that your biology teacher has assigned you guys in regards to passive, active transport, or they might have also named it cell transport. Okay. Have a great day, guys, and we'll see you later.